Evening. Good evening and welcome to Evening at Egan. Can you guys hear me? Hello out there. There we go. Welcome, and I'm so glad you're here, and it's an enthusiastic crowd right from the beginning, so you've come to the right place. I'm Marcia Souza. I'm the Dean of Arts and Sciences here, and it's always a pleasure to welcome people to another evening at Egan, and especially when one of our own faculty is speaking. There's actually two presentations tonight, so I do want to clarify that because JWAC is also sponsoring a presentation on China in the library. So if anybody's in the wrong place, go ahead and stay here because this is going to be awesome. And before I introduce Dave, I do want to remind you about next week's presentation, which is UAS in Cuba, a semester-long search for Che, Hemingway, El Papa, and the authentic tourist. Last spring, 4 of our faculty members and about a dozen students spent 10 weeks in Cuba, and they're going to share photos and vignettes and stories and tell you about their kind of life-changing event last spring, so that'll be good. But tonight our speaker is David Tallmon. He's an associate professor of biology here at UAS. Dave earned his PhD in organismal biology and ecology at the University of Montana. And he's done research in England and in France, and he just got back from New Zealand where he did a sabbatical, and of course he's done a whole lot of research right here in our own backyard. His interests are in evolution, ecology, and conservation biology. He works on a huge variety of species. He'll tell you about a couple of them tonight. Salmon and sculpin. He works on seals, toads, grouse, crabs. There's a long list. So I think if you provided him with something curious, he'd find a way to work on it. Tonight he'll be discussing his recent work on salmon and sculpin in Southeast, and you can see the title of his talk right up there. So there will be time for questions afterwards, and we'll have a microphone to pass. We are filming, so if you want to ask a question, wait for the microphone. And for now, help me welcome Dr. David Tallmon.
Thanks, Marcia. So I'd like to talk tonight about some of the work that I've done along with some students and collaborators in Auke Creek. And in particular, I'll talk about a couple of fishes. We'll talk about salmonids and also sculpin that use Auke Creek. Before I get started, I want to make sure to give credit to a lot of folks who've contributed to this work, some of whom are still around and some of whom are recently departed from Juneau. Ryan Kovach is a PhD student of mine who just finished up a couple weeks ago at UAF. Andrew Whiteley and Carolyn Bergstrom, these two here, were postdocs in my lab who have moved on to professorships at UMass, and Carolyn's at UAS. And Tyler Linderoth, who was an undergraduate here at UAS, who's now doing a PhD at UC Berkeley on population genomics of sexual selection in tropical frogs, if you can believe that. And then also John Joyce, who appropriately enough in this picture is being splashed by a salmon in the holding pen in Auke Creek. Sorry, John, that's the best photo I could come up with for you on short notice. And then Tony Garrett, who is a longtime faculty member in fisheries out at UAF. So all these folks have contributed to various aspects of this research tonight. It's by no means my own, but very much a collaborative effort. Okay, as many of you know, Alaska is experiencing some pretty radical, rapid changes in the physical environment. This is an image here that I just pulled down from the internet today. It shows the extent of the Arctic polar ice cap as of yesterday. And shown here in white is the extent of the ice cap. And then shown here with this colored line is the median extent of the ice cap from 1979 to 2000. So pretty strong evidence that the ice cap is shrinking. I'm sure you've heard about it on the news. This figure over here shows the extent of the ice cap over time. So these are the months when it reaches its smallest extent. And this shows the average right here from, 1979 to 2000. And then shown here down below is 2007 and 2012. So pretty strong evidence that we're seeing some shifts in the physical environment. If we look a little closer to southeast Alaska. This is a study that came out in 2002 looking at glaciers throughout Alaska. So the glaciers were broken down into various regions, numbered here on this figure in the upper left. And if we focused on southeast Alaska, right here, number 3, our region right here, also shown right here, what you can see is that there's a net loss shown below this x-axis here for glaciers in southeast, and they're being lost fast, and in fact, the rate of loss is accelerating. So increasingly rapid change in glaciers. And obviously, changes in glaciers are going to have an influence on the fishes that those glaciers melt into the streams of, and then also ultimately into the ocean as well. Okay, and then bringing it back really close to home, this shows temperatures in Auke Creek. So here's the holding pen at the weir for Auke Creek, and you can see here the temperature, the year here on the x-axis, and then the mean annual water temperature over time. And changes in temperature are a concern with fishes because fishes are poikilothermic, or most of them are, so they change their body temperature with the ambient environment. So we expect that with shifts in the environment, you'd see shifts in their metabolism, developmental rate, and those sorts of things. Okay, and as I mentioned before, I'm going to talk primarily about 2 groups of fishes. So Sculpin, shown here in the lower left, and in particular the Coast Range sculpin that uses Auke Creek and many streams up and down the west coast. And then I'll also talk about salmonids, including salmon, char, and cutthroat trout that go in and out of Auke Creek and go by the weir where they are counted. So the question here is what's happening to these fishes as temperatures change? How are they responding? And you know, if you're a terrestrial species or marine species, you can shift north in latitude as temperatures warm, right? So in the northern hemisphere, you'd expect things to move north, and there's patterns for some species that they're actually doing that. But in the case of stream-dwelling species, or rather populations, they can't move north. They're constricted to the streams they're found in. And on a local level, our concern is at the population, because those populations either are there or they're not, even if as a species something moves north. So if you're a commercial fisherman or a subsistence fisherman, if salmon move up to the Beaufort Sea, that's of little comfort. What you want to have is salmon in your local stream so that you can harvest them. So really what we're concerned with freshwater fishes, or fishes that use fresh water, is whether they're going to evolve in response to a changing environment or die. Those are really the 2 alternatives. So let me start talking about Coast Range sculpin, shown here. So this elegant creature is a bottom-dwelling freshwater fish, although it's amphibious. So for part of its life cycle, it actually goes into the ocean as larvae and then swims back up into the streams. It's known for its cryptic patterning, so its ability to hide itself. It's also known for quickly inhabiting or colonizing streams as glaciers melt. And it's also a pretty voracious predator of salmon eggs. So when the salmon come in, the sculpin are happy. They follow up behind the salmon and gobble their eggs up. So initially, our studies of sculpin were done in Glacier Bay. So Glacier Bay is shown here in this map. I'm sure a lot of you have seen this picture before. And you may or may not know that Glacier Bay is world famous for looking at glacial succession. So as glaciers recede and the land changes over time, Glacier Bay provides an ideal natural laboratory for looking at those phenomena, because there's a really nice record or chronosequence of changes in the glaciers over time. So this shows where the glaciers were back in the late 1700s to the 1800s, to more recently. Sorry, this thing's really awkward. To more recently. And what we noticed as we moved along this chronosequence, or these streams that were slowly uncovered as the glaciers receded, is that there was this incredible pattern in the sculpins, the Coast Range sculpins, that they seemed to change in color as you went from older streams to younger streams. So in Glacier Bay, that's going from older to younger streams. And another interesting aspect of this is that the fishes definitely matched the background of the stream they were found in. So the older streams tend to have greater development of algae, more red and green colors, and the sculpin in those streams seem to match that background. If we went to the newer streams, the ones that are more recently deglaciated, we found a lot more white coloration, and then the sculpin also matched that background that they're found on. And so this suggests, one, that sculpin are able to match their background, and the question was, is this an adaptive, genetically controlled coloration pattern, or was it a more plastic or malleable trait, so that basically you could take a sculpin from anywhere and it would adjust its color to match its background, or was it this genetically based and had to evolve across generations? And obviously, matching your background is an important thing if all of your predators have good color vision, in the case of bird, fish, and mammal predators of coast-range sculpin. So we first asked the question, is this coloration plastic or is it genetically based? And to do that, we took sculpin from across the chronosequence in Glacier Bay, old and young populations, and moved them into a lab and put them on a common background, and then basically tried to push around the coloration of the sculpin by changing the background that they found themselves on. So this figure here shows time in days, and it shows coloration here. So it turns out coloration's a really complicated trait. But we've sort of reduced it down here to a single axis, and going from dark to light. And essentially what we found is that if we took fishes from different places, so shown here is OY is for Oyster Catcher Creek, for those of you who've been to Glacier Bay. It's a little lower down in the bay. And then Wolf Point Creek, which is a little further up the bay, so a younger population. And we moved the fishes onto a common background and then changed them onto white or black backgrounds, and then moved them back to a common background. And essentially what we found is we could take the phenotypes or the coloration of those fishes and cause them to diverge or converge no matter where the fishes were from. So again, this suggests that it's a highly plastic trait. So the next questions are, well, okay, so how much does this matter that they match their background? And then if there's selection for them to match their background, natural selection by predation causing them to match their background, if their background is changing more rapidly, as we see more rapid deglaciation, is the ability to adjust to your background a costly trait? So does background matching affect survival? To look at this, Tyler Linderoth, the undergrad I showed you, and Carolyn Bergstrom devised an ingenious pseudo-sculpin. So they made plasticine sculpin that were either light and dark in color, and they put them on light or dark trays, and they quantified the divergence between the model fish and the background it was found on, and put them out in Auke Creek. And they asked the question, do the fishes that differ more from their background have a higher rate of predation than those fishes that are a closer match to their background? And so you can see here, these are different fishes, different light or dark fishes on light or dark backgrounds. And you can see in this picture here that in fact things actually do prey upon model sculpin. So they fooled even the predators themselves. And there was also a very strong effect of background matching, suggesting that there was a strong effect of background matching, and the more the fishes diverged from their background, the more likely they were to be predated. So again, this confirms what you might expect, but it's an experiment to demonstrate that background matching is an important trait for these sculpin. So strong natural selection to match your background. So the next question that I mentioned before was, well, okay, they can match their background, but is it costly to be able to track your background? So you think about the fact that these sculpin are able to rapidly shift their coloration. That takes a lot of energy. They've got to differentially kill or cause the proliferation of different cells that give them different coloration patterns. Is that costly for those Sculpin? And so again, we took Sculpin from Auke Creek and moved them into the lab and either kept them on a control background, a lighter dark background that wasn't changing over time, or put them onto a lighter dark background that we changed either very rapidly on the course of a couple of days or more slowly on the course of a couple of weeks. And then we measured them over time and asked, is there evidence for a cost to this plasticity or having to track a changing environment in terms of their growth rate or their survival? With the prediction that as the environment changes, that's going to be costly and it's going to reduce either growth rate or survival. And this is what we found. So for growth, we didn't see any effect. But what we found for survival is that those fishes that were moved onto a slowly changing background, so changing on the order of weeks, they actually had reduced survival relative to the controls, suggesting that under some conditions, for some traits, there can be a cost to this plasticity or this tracking of the environment. So this is the treatment, so control, fast change, slow change, and this is time elapsing here, showing the differences in survival with the lowest for the slow change. So for Sculpin color variation, we found first that they're very plastic. We found that background matching was important for survival, at least for our model Sculpin out in Auke Creek. Presumably, you could extrapolate that to real Sculpin in Auke Creek and elsewhere. And we also found that there was evidence that survival could be costly in terms of one of the traits we looked at in the lab. So again, suggesting that Sculpin have this amazing ability to track and adjust to their changing conditions, but under some conditions that we were able to look at, it looked like there could be a cost to being able to do that. Okay, so I want to shift now from Sculpins to starting to talk about Salmonids, and I'm also going to focus, instead of coloration and cost of coloration, I'm going to look at phenology. So phenology is really just the timing of seasonal events that occur on a regular basis in the life history of an organism. And perhaps you can think about it with this, this hair here. So hairs change their color from light to dark, and natural selection should act on those hairs to be white in color when there's snow on the ground, or to be dark in color when the snow is melted. And if you're a hair that's still white in color when the snow is melted, or if you haven't quite changed your color when there's snow on the ground, that could be costly in terms of survival. And so the idea here is that natural selection acts on organisms to cause them to track their environment, and those individuals that track the environment better leave more offspring or survive better, and there's selection to closely match your environment. So the same thing should hold with salmon or salmonids. They have this fairly complex life history that occurs, and we expect them over time to adapt so that the events in their life cycle match optima for migrating, for laying eggs, those sorts of things. And one thing that I want to emphasize is that salmon are also known for their incredibly diverse life histories. So across species or across populations within species, there's these genetically based traits that vary a lot. And they can vary in all these ways that are shown here. So they have variable numbers of years that they spend in fresh water. Some may go to lakes for some of that time. Some may go to streams for some of that time. And they go out to sea. They vary in the number of years or months they can spend in salt water. You also have this variation, genetically based variation, in the form of early and late runs within populations. So you'll get a pulse early in a season and then another pulse later in the season from the same group or species in the same stream. There's also odd and even year runs. So in the case of pink salmon, they have a strict 2-year cycle, but they have because they have a strict 2-year cycle, there are odd and even year runs within a given stream that are genetically and demographically independent from one another. And then there's also a lot of variation in the form of things like jacks. So jacks are basically like teenage males. They go out to the ocean and come back early to spawn rather than reaching full size of most adults. And it's thought that all this variation that exists within salmon species and populations is a buffer against unfavorable environmental conditions. So it's called biocomplexity, just a lot of variation, genetically based variation that provides a buffer against changes that might occur in the environment. Okay, and what I'm going to focus on today is salmon migration timing, and some things to know about it. It has a genetic basis to it. The amount of genetic control varies among populations and species, but there's good evidence that it has a strong genetic component to migration timing. There's also evidence even from Auke Creek and work done by Tony Garrett showing that these species and populations can be locally adapted to thermal conditions. And I don't need to tell anybody in this room that they're economically, culturally, and socially important. So lots of people depend on them here in Juneau. Cultures have made them a strong part of their identity. And of course, economically, we know in Alaska there were hundreds of millions of dollars annually. And then again, so the question is what's happening in Auke Creek as we see these increasing temperatures? How are the salmonids responding? So we're fortunate in Juneau that we have the Auke Creek weir. It, as far as I know, is unique on the West Coast. You have a bunch of wild populations of salmonids that for many decades have been completely censused. So everybody coming in and everybody going out has been counted one by one for decades and decades, thanks to the heroic efforts of people like Jerry Taylor, who used to be at NOAA, and John Joyce and others who are carrying on the work. And in Auke Creek, there are coho, sockeye, pink salmon, dolly varden, char, and cutthroat trout. And the reason we got interested in this was because some of the work that Jerry Taylor did before he retired suggested that the pink salmon in Auke Creek were adjusting their timing, their migration timing to earlier in the season, both as adults and juveniles. And we were also interested in looking across the suite of species and then focusing specifically on pink salmon because thanks to Tony Garrett, there were genetic data that had been collected going back all the way to the 70s that we could continue on and look at evolution in pink salmon because we had genetic samples in this case. Okay, so this figure shows the phenology of the salmonids in Auke Creek. So again, the timing of their migration. So on the left side here is migration from freshwater to saltwater, migration from saltwater to freshwater over here. And you can see the various species here as they're migrating out or migrating back in to Auke Creek. You can also see, shown here on this axis is temperature, and shown here is the average from 1971 to 80, and from 2002 to 11. So you can see also that temperatures have increased, in particular in the spring. Okay, so our first question was, is migration timing changing across all the various species and life histories that we have in Auke Creek? And to do this, we use what's called an information theoretic approach to look at changes in the median date of migration timing, so the midpoint of when they migrate, and also the range of dates when they migrate, so the time between when the first fish come in and the last fish come in or migrate out for each species or life history type. So again, just we're looking at these two parts of the life history, smolt migration, or when the juvenile fish are heading out, or spawning migration, either what we're calling the adults or the fully mature individuals, or jacks, which are the young males that come back from the sea early, not at full size. we were also interested in looking at a suite of variables that we thought might explain the variation in the timing of the adults and jacks, and also the juveniles, as well as trout and char. Okay, so this is what we found. So we predicted, perhaps naively, but perhaps not so naively, that migration would be earlier as temperatures warmed, that the trend would be toward earlier migration. And if you want to remember just one thing from this slide, for 11 out of the 14 species in life histories combinations, we found there was earlier migration. And I'll go into more detail on the figure now. So below this axis right here is the species for which there's earlier migration. And you can see this key down here at the bottom. Different species have different colors. And then the different life histories, so looking at adults or jacks, have different patterns for each color. And the figure's broken down to, on the left side, migration from saltwater to freshwater, and then on the right side, freshwater to saltwater. So again, everything here and below is earlier, and everything here and above is later over the course of the period of multiple decades that we're measuring migration timing. So some things to note. For most things, as I mentioned before, they're coming back earlier, but there's also some variation even within species across life histories. So this shows for sockeye, these are the jacks, which are coming back earlier. The adults are coming back later. Shown here are sockeye juveniles going out and coho juveniles going out. In this case, age 1 sockeye, that is, they spend 1 year in Auke Lake before heading out. These are age 2 sockeye, spend 2 years in the lake before heading out. Similarly, age 1 coho and age 2 coho. Interestingly, the age 1s and age 2s in these two species showed different patterns. That is, the age 1s are coming out later, the age 2s are coming out earlier for both of those species. But in both species, there's fewer and fewer age 1s over time. So overall, there's a shift towards earlier migration, even though there's this difference between the age 1s and age 2s. So if you prefer to see things in text, these are some of the highlights from what we found. So looking at the adults and jacks, odd year pinks, which are demographically independent of the even year pinks, coho adults, coho jacks, sockeye jacks, those are all migrating earlier. The exception was the sockeye fully mature adults. The extreme was the coho adults that are now migrating 17 days earlier than they did 40 years ago when these data began to be collected. Everybody except for the sockeye jacks appears to avoid high stream temperatures and low flows. If we look at the juvenile salmon, dolly varden, and cutthroat, everybody was earlier except for the age 1 coho and sockeye. But again, there's fewer of the age 1s over time. The extreme in this case was odd year pink juvies, which are migrating 19 days earlier than they did 40 years ago. Some other interesting facts that came out of this analysis, dolly varden and cutthroat migrate about 9 or 10 days earlier for each 1 degree C increase. And pink juveniles migrate 7 days earlier for 1.33 C increase. So this shows the patterns in the median. We also were interested in looking at the range. So again, the range is basically the length of time over which the runs are occurring, shrinking or increasing over time. We predicted that if there's directional selection, so selection towards earlier migration, there would also be evidence of selection in the form of a reduction in the range of time over which they occur. So one of the things selection does is it reduces variation. So we predicted that there would be a change in the range, and it would be towards a shrinking range, and that's what we found for 11 out of 14 species. Again, this is the same sort of figure. I know it's ridiculously complicated looking, but again, we have the different species with different colors. The life histories show different patterns. We've got migration saltwater to fresh, fresh to saltwater, and below the axis are narrower migration ranges, and above the axis are wider migration ranges. So we're seeing shifts in the median. We're also seeing shifts in the range. And another striking pattern that we saw was that if you look across the range of dates over which mature salmon are coming back from the ocean, that period of time has also shrunk over the time series. So that if you count from when the first 100 fish, adult salmon, come in to when the last 100 salmon come in, the range of dates over which that occurs has shrunk over time. And we looked at this multiple ways, looking at the number of days the adult salmon pass by the weir, all sorts of ways. No matter how you slice it, essentially the range of dates over which these salmon are available, either for consumption by fishermen down at the mouth of Auke Creek or by non-human vertebrates, bears, mink, whatever else eats them, the range of dates over which they're available is shrunk. And that's obviously of concern for those of us who want to catch them. But the good news in all of this is that even though we've seen shifts in their migration timing, we see decreases in the range of dates over which they're available, their abundances appear to be stable. So this shows for even year pinks, odd year pinks, coho, and sockeye. On the y-axis is a log of abundance, so it's just a transformed abundance. On the x-axis is time, again. Essentially what this shows is that all these populations are not different from stable. So if you look at this is lambda, which is population growth rate. Population growth rate of 1 is stable. And you can see looking across all these population growth rates, and in particular the confidence intervals, they include 1. So these populations can't be distinguished from being stable over time, which is good news. So to summarize the Salmonid migration timing, 11 out of the 14 species in life history show earlier migration. 11 out of 14 show narrower range of dates over which they're migrating. And even though they appear to be less available in terms of their temporal availability to humans and everybody else that likes to eat them when they come back from the ocean, their abundances are stable. Okay, so that's the general picture looking across all the different salmonids in Auke Creek. We also wanted to focus in on pink salmon because thanks to Tony Garrett and some others, there's been long-term studies done of the genetics of pink salmon, showing evidence of local adaptation in pink salmon. And also there are genetic samples that were available for us to analyze looking across time. And with the availability of genetic samples, we could look at evolutionary changes or microevolutionary genetic changes that occurred in pink salmon. So one of the things that Tony Garrett and his collaborators showed is that there's evidence within the odd and even year runs of local adaptation based on migration timing. So that within pink salmon in Auke Creek, they looked at early runs shown here. So these are dates here and then abundance here, that there was a distinct early hump and then a later hump in this case, odd year pink salmon. So the salmon that come back on odd years. And they did crosses and shown that there was genetic basis to these two humps and thermal adaptation in terms of their developmental rates. And also thanks to Tony Garrett, we had a genetic marker of the late run that was available. So back in the late 70s, they bred a genetic marker into the late run of that second hump that I showed. And this is evidence of that genetic marker. So this shows for 85, 87, 89, that there's a genetic marker that shows up late in the return time for these pink salmon. So this is the frequency of this genetic marker. You can see it's fairly low frequency early in the return, and then when the late run comes in, it reaches a much higher frequency. And this marker was stable through the 80s. But it turns out 1989 was the second warmest year on record, and Coincident with that was a loss of that genetic marker. So if you look in the subsequent time periods, 91, it looks like that marker no longer achieves a high frequency late in the return, and that continues through time up to last year, 2011. So essentially that genetic marker and the fish that had that genetic marker were lost and didn't leave progeny behind. Fortunately, we also have a genetic control. So this shows essentially the same thing, but with a genetic control. So this is this marker allele shown here in dark over time. So here's the frequency of this genetic marker in the population as a whole. This is year, and you can see that there was a frequency here, bounced around, and then a loss of that late run marker. In contrast, this other gene that wasn't a marker for the late run, but rather common to the whole population, has not changed in frequency over time. Shown here, frequency of about 10%, and still at a frequency of 10%. So this suggests an evolutionary change in the odd year pink salmon in Auke Creek. So in terms of selective forces, we don't know for sure, but we know that 1989 was an abnormally warm year. We also know from the work of Tony and others that early migrating fishes are better adapted to warmer years, and that in 1989 we saw the loss of this late run. And you can see here, looking across from the start of 1971, when they first were collecting data, to 2011, you had the two distinct humps. By 91, the two humps were gone. And 2011, you see a shift in the migration timing, so the percent of returning adults towards earlier, but also that late run didn't come back. So that's evidence of there being natural selection against the late run. But the good news, again, is that the population has remained stable. So even though the late run was lost, there was evidence of some decrease in the population in the subsequent returns. Since then, the population has completely rebounded, and last year was the second highest number of pink returns for the odd year population. So it looks like in this case there's been a cost in terms of a loss of biocomplexity, or the late run of this population, but the population as a whole is still abundant as it was when they first started collecting data 40 years ago. Interestingly, this same pattern of early and late runs within a year, so this is odd year pink salmon, even year pink salmon, and remember, for pink salmon, they're locked into a 2-year cycle, so the odd and even year runs are demographically independent of one another, don't influence one another. They both had early and late runs, looking back in the 1970s, so early 1970s, this is 71, 72, you can see the two humps here. In the case of the odd year, that was lost sometime in the late 80s, 89 we think, and didn't come back. In the even years, it was still present by 1990, but lost sometime between then and now. So we see evidence of a loss of biological complexity, or these genetically based parts of the pink salmon run, but overall, the pink salmon are still abundant. So if I could summarize here, looking across these Auke Creek fishes, we see evidence of these extremely plastic sculpin, an incredible ability to track their environment in terms of their coloration, their crypsis, their ability to hide from predators, and some evidence under certain conditions that that ability can be costly energetically or in terms of survival. When we look at the salmonids, again, we saw this shift of 11 out of the 14 species life history combination shifting to earlier migration timing, and same number showing a narrower range of dates over which they're migrating, which is consistent with natural selection, although we don't have genetic data for anybody except for the odd year pinks. And then, of course, the upshot of that is that we have reduced temporal availability of these pink, or of these salmonids for harvest, and on the flip side, they still seem to be just as abundant as they ever were, or at least when we started collecting data. And then in the case of the odd-ear pink salmon, we actually have genetic data suggesting evolutionary changes in these pink salmon, and the loss of this late run back in late 80s. But again, abundances seem to be just fine. So taking a step back and thinking about this, I guess the first thing I should state is that the big caveat to interpreting this is that we've got a sample size of 1 in a sense. We've looked at sculpins in Auke Creek, and we've looked at salmonids in Auke Creek, but it's only one stream, and these species range over a vast geographic area. So we're really fortunate that we have the Auke Creek data sets available, but it's one data set in a sense. So it's important to be somewhat circumspect. But I think it provides some really encouraging results in the sense that it shows the great resiliency of these fishes. They're highly fecund, so incredible ability to make babies really fast, which allows them to respond to changes in their environment. They're not like seals, ice-breeding seals, where they have a pup a year, or a polar bear that has a few cubs every year, and if the conditions change, it takes them a long time to rebound. These things have an incredible capacity to rebound. And that's reflected in the fact that their abundances are stable. We're also fortunate in the sense that Alaska salmon have it fairly easy, right? It's not like the fish down south. There's not huge impacts from the 4 H's, hydropower, hatchery, habitat, and harvest. And we have harvest here, but we've also got our habitat largely intact, and we don't have that much influence from hydropower or hatcheries. So in a sense, Alaska fish have fewer stresses on them, which is going to allow them greater ability to respond to the stress of climate change. The darker side of this is that the future is uncertain. So it's unclear if this resiliency they've shown in terms of their ability to change their timing, yet still show high abundances or population growth rates, is going to be true in the future. The predictions from the physical models are that not only is the mean temperature going to increase, but also we're going to see incredible increases in the amount of variance around that mean. So we're going to see greater and greater oscillations over time. So it's unclear if their ability to track things the last 40 years reflects what they're going to do in the next 40 years. And then there's also the important point that these salmon don't live in isolation of other species. So they've shown a strong ability to respond, but they've timed their migration to take advantage, say, of the plankton that are blooming in the ocean. And if the plankton aren't blooming in the ocean at the proper time the salmon have adapted to, then that's going to have an impact on the salmon as well. So it's important to not think about these species in isolation of the species that they eat and that eat them as well. And how they're going to respond to climate change is also going to influence the success of salmon. Okay, I want to make sure to acknowledge sources of funding. So this work, in addition to the great people who have worked on it, has been supported by some generous agencies, including the North Pacific Research Board, Alaska EPSCoR, Alaska Sustainable Salmon Fund, National Park Foundation, and the International Polar Year. And I'll just leave this up. If people are interested in reading more, there's some amazing books that have come out in the last few years by David Montgomery and Tom Quinn that might be of interest to you. And then I also leave reference to some of our papers that show these results in greater detail if people are interested in those. And I think with that, we've got time for questions. So thank you.
Thank you. You indicate that this work is all based just on Auke Creek, on fish in Auke Creek. Are you aware of any work that's being done to try and generalize these results to other streams, like say the Mendenhall River, which services similar species, or others?
Yeah, so I'm actually working with some people to try to take advantage of WEIR data throughout Southeast Alaska. So there are a number of streams throughout Southeast Alaska where there are WEIR data available. The unfortunate thing is that a lot of the data from elsewhere aren't continuous, so there are huge breaks in the data sets, and they don't have permanent WEIR structures that catch everybody. So they might start a little bit late some years, or maybe have to end a little bit late some years. They might have blowouts. So there really isn't, as far as I know, a data set that's as good as Auke Creek anywhere, certainly in Southeast. But there are data sets that are somewhat comparable that we can probably look at and may provide some insights and ability to generalize. And that's actually something that I'm working with Sanjay Piari and other folks at UAS and elsewhere on. So hopefully we can make greater generalizations.
Thank you very much. You mentioned the abundance, like for the pinks, for example, is the same, but are we seeing any variation in size over this period of time?
You know, I don't have a very good handle on that. So I can't answer for Auke Creek very well. I know that there's pretty good evidence throughout the range of salmon that sizes are shrinking most places. But I can't answer for Auke Creek. We haven't looked at those data, although I think we've talked about it. But we haven't done it, so I can't answer. I'm sorry.
Great, thank you.
Thanks. A couple of those results seem sort of counterintuitive to what I would have expected. And a couple questions. Do you have any idea of the mechanism of the mortality in pink salmon that sort of favored the earlier runs as opposed to the later?
Yeah, so we have some indication. So there's some data that suggests that the late year offspring from that year that were really high temps, it looks like they had much higher marine mortality. So the guess is that they didn't time it right getting out to the ocean and suffered higher mortality once they got to the ocean.
And the declining frequency of year one fish as opposed to year two also seems a little counterintuitive.
Yeah, I agree. We haven't completely gotten our heads around that one. So I can't answer that definitively. There must be something that's telling them it's good to hang out a little longer in fresh water now. And I can't really come up with a good scientific reason why that is. But yeah, it's kind of perplexing. If you have a really good hypothesis, let me know.
Thank you for the excellent presentation. I have a question about Sculpin.
Good, thank you.
You talked about the color variation and the fact that these are primarily fresh water. So the question is, in an area like Glacier Bay, how do they get to these new streams, and what's the exploitation? Or not that, I mean, how do they discover, and how long does it take? How do they get there, basically?
Yeah, so it's pretty amazing. We've also done work on their genes, and it appears they have an incredible ability to essentially go everywhere really fast. And what we think is going on is they hatch out in fresh water, float down into the ocean, and then they're planktonic for a period. And apparently they get mixed well enough by the ocean so that we see that they're essentially genetically identical throughout all of Glacier Bay, even though they show this morphological or this difference in their coloration. So it's simply planktonic dispersal that leads to these differences, because that's the stage when they're in the ocean. After that, they're sitting tight in the streams. So it has to be that period.
Are the salmon recolonizing the recently deglaciated streams before the Sculpin, or what's the time?
Yeah, good question. I think it varies from stream to stream, and I know there is evidence in some places they have colonized before Sculpin. So I know of at least one stream in Glacier Bay where the salmon have gotten there before the Sculpin, but I don't know if there's any general knowledge of, or general patterns that are known.
I was wondering if there's been any analysis of whether or not the returns are related to the hatchery or not.
Yeah, are you thinking just in general for the salmon, or?
Well, since you've got variation in early returns, is there any correlation between non-hatchery and hatchery fish?
Yeah, so we're fortunate also in Auke Creek that there's very little hatchery influence. And there have been some years where there has been hatchery influence, and we teased that apart statistically. But for the most part, there's been very little hatchery influence on Auke Creek. So that's another great thing about it, is that we can, unlike a lot of places down south where there are weirs, there's also a huge hatchery influence. So it's hard to know how much of that's a natural pattern versus selective pressures that have gone on in the hatchery. And we don't think that's a huge issue in Auke Creek.
So 2 questions about the Sculpin. You mentioned that they were eating a lot of salmon eggs. Is that a good thing or a bad thing?
Well, if you're a Sculpin, it's a good thing. I think in Auke Creek, it doesn't have a huge influence on the salmon. I think it's mostly just sort of slop and spillover that they're getting for the most part, is my intuition. But they may have it. I don't think they have a huge impact on the salmon populations. Is the way I would answer that.
So second question would be the hits on the plastic Sculpin. Is that mostly fish, or is that birds, or what?
You know, there were a lot of cases that we couldn't identify exactly what it was, but there were definitely birds and mammals and what we think were fishes that hit them. But there was a huge proportion of the cases that didn't have the clear bite mark that I showed you. Now, that was sort of an idealistic strike. But there are a lot of cases we don't really know what was getting. We assume it was all 3, and there's evidence certainly of what looked like bird pecks and also mammals dentition. So.
How do you count the fish as they come in through the weir? And has that changed over time?
Well, there's basically a blockage. So there's a mesh, if you will, that's put down that they can't slide through. So they are essentially funneled into a holding area where they can be counted, and then
People?
passed by people. Yeah. So it's a human endeavor. It's not an automated. Thank you. Automated process. It still requires humans to do that part, thankfully.
Thank you. Department of Transportation has some plans to make some pretty big changes along the road up above Auke Creek. And I'm wondering if you've looked at all at what might happen with retaining walls and loss of trees and how that might impact anything.
I personally have not looked at it at all. I know that there has been some work done looking at some recent telemetry work, as well as sort of uncovering of past telemetry work to try to see where the coho and sockeye in particular hold before they spawn in the lake, and see if there might be some impacts from those developments on them. But those data were just collected this summer, and so I don't think they've been analyzed or if there's any definitive picture from that. But you should talk to the Fish and Wildlife Service. Our local office are the ones who are, I think, leading the charge on that.
How do you do a count of the outmigration for pink salmon?
How do you account for it?
How do you do a count on?
Well, there's the weir structure actually blocks everybody from going out, and so you can count you count them as they go one by one. So you've got a blocking mechanism that you essentially set in one direction to catch everybody as they go out, then you flip the structure and catch everybody as they come back in. And
I thought
count
they
them.
were incredibly small.
Well, they are.
Great.
They are small, but they're not. You can see them with the naked eye or whatever. Yeah.
Thank you.
Okay, I think that's it. Thanks a lot. I appreciate y'all coming out.